let's continue. So we're going to look at some specific crops, some specific examples, and kind of put the science and the food and all of this together. So the first one is golden rice, which is a very popular, well-known example in a lot of parts of the world. Here we have regular white rice, and here we have our golden rice expressing large concentrations of beta-carotene, which gives it the yellow color. Okay? So golden rice is a variety of rice, so it's a regular rice product, and it has been produced through genetic engineering to synthesize beta-carotene, which is a precursor of vitamin A, in the edible parts of the rice. Okay? So this research was initially conducted with a humanitarian goal, with producing a food that was fortified and grown in areas of the world that had very much a dangerous shortage in dietary vitamin A. And a deficiency in vitamin A is quite significant, mostly for children and during pregnancies, and it's estimated to contribute to the death of 670,000 children under the age of 5 each year due to severe vitamin A deficiency. It also contributes to millions of causes of blindness. But that will be a severe deficiency where they're not obtaining anywhere close to the recommended daily allowance. Okay? So globally, when we talk about the prevalence of vitamin A deficiency, when we look at the map, and I'm just going to point out the red areas because that's really what we're talking about in terms of the research and where it's grown. So large areas of Africa and India and some parts of Southeast Asia. Green is least severe. Green, of course, also looks like most of the Western world in terms of the developing world. Countries in blue, so you see, whoa, there's like big parts of Europe in there, are data that has not been reported significantly, so we don't have an overall. But most of Europe, all of Europe, really would be considered not a problem area, even if we looked at the far places right down here along the border with Russia and old Soviet Union countries. They still are not considered countries with vitamin A deficiencies. So the difference between golden rice and regular rice is the addition of these beta-carotene genes. Okay? And it was first published in the year 2000, so it's a pretty new crop. And they came out in 2005 with a newer variety called golden rice 2, which produced up to 23 times more beta-carotene than the original golden rice. So even though they produced it initially, they are still working on improving it and continuing to do it today. They really are working on trying to produce a more nutritious rice product. So initially, the goal was to develop it as a humanitarian tool to produce a food product that could be grown in the areas that it was needed. So a lot of the controversy and more like socioeconomic discussion about GMO foods, we're producing GMO cotton and corn and soybean, and these are grown in parts of the world perhaps and then exported, or grown in parts of the world where they're not needed the most for food. So the idea of golden rice would that we would be able to produce a food product that then would be grown locally for the people that actually needed it and were grown rice anyway. So they would grow rice for a portion of which they could keep for their family, and then a portion of which would be bought and exported. So the idea was that the families, very poor families who were growing rice, would be able to grow one crop and that it would provide increased nutritional value, okay, because they were already eating rice anyway, right? So the goal was, it was a great goal for humanitarian reasons, and even though that's how it was introduced, it has met with significant opposition from environmental groups and anti-globalization activists. And some of these groups, are smaller groups and may not have such big influence, but large groups like Greenpeace, for example, which is a very big global presence. A lot of people read what Greenpeace says. They partially agree with what Greenpeace says. They may really look to them for good data, and Greenpeace did really not agree with golden rice, not because it was a humanitarian tool, but they really didn't agree with the genetic engineering portion of it. So some countries were disappointed in that because they felt that Greenpeace has a far-reaching influence, and a lot of people listen to what Greenpeace says. I mean, most people know what green peas is, and they understand that they try to do good things. So a lot of groups, political groups were disappointed that Greenpeace came out and kind of disagreed with this. Okay. So that's kind of that's one side of it. Studies have found that golden rice poses no risk to human health, and multiple field tests have taken place with no adverse side effects to participants. This in itself created quite a controversy because a lot of the early studies with golden rice to see for safety were carried out in China with Chinese children. So they were carried out with children straight away, eating golden rice, as part of their diet. So there was a lot of objection to this because we were using children in a trial as pretty much guinea pigs, whereas if we were treating this as a medical supplement, which is how some people view it, we wouldn't go straight to children. It would be tested elsewhere more thoroughly first. So there was a lot of problems with those initial trials. The initial trials were supported by the Chinese government, but after the kind of international disappointment and outcry came about, Chinese children being involved, then the Chinese government stepped back and said, oh, we did not support the scientists, and then the scientists who ran the study were impacted in terms of they were blamed for setting up a poorly designed study. So, I mean, this has happened many times throughout recent history. It's nothing new, but it's definitely controversial. However, the data that was collected in terms of nutrition showed that there was no negative impact in terms of using golden rice in the diet of children. Oh, that was the round 2. 2000 was they first brought it out. Oh, they were right. That was in like the 5 to 7 years prior. Mm-hmm. Yeah. So it's still yes, but still we would still consider it new. Yeah. Mm-hmm. So how do they kind of set this up? So beta-carotene is produced in the endosperm of the plant, which is the edible part of the rice. And it's a very important precursor to vitamin A. And if we think earlier before the break, I said that, we can control where in the plant the enzyme or the protein or whatever is produced. So this was one of those mechanisms. They designed all the beta-carotene to be produced only in the endosperm, not in the remainder of the plant where the whole rice plant grows. So they concentrated the beta-carotene in the part that would actually be eaten. So that was a well-organized design in terms of where is the gene going to be expressed. It was only going to be expressed in that part. The rice plant then naturally produces the gene products in its endosperm. In the normal environment, rice also produces a small concentration of beta-carotene in its leaves. And that's important for photosynthesis. All plants produce a small concentration because it's required in one of the cycles involved in photosynthesis. However, we would need it specifically in the endosperm because that's where we eat the part of the rice. So that's where they worked on trying to get it to be produced just in the endosperm and increase how much is being produced. How they did that was they used a bacterial gene. It's called phytene desaturase gene. It's a bacterial gene. It's abbreviated to CRT1. And this actually comes from a genetically modified tomato because in the tomato, this gene produces lycopene from phytoene. So lycopene is the nutrient in tomatoes that's very it's important, healthy part of that whole process, a very important enzyme. So in tomatoes, this enzyme has been inserted into some genetically modified tomatoes, even though it also naturally occurs, the lycopene part, and it converts phytene into lycopene. So what they did is they used this process and inserted it into the rice so that the enzyme is now in the rice, a bacterial enzyme is in the rice, and it produces lycopene in the rice, and then lycopene is converted in the rice into beta-carotene. So the beta-carotene is produced de novo, which is newly, in the rice itself. Okay? So they got around trying to let's put more beta carotene in there, let's put this gene in there to increase expression, let's change the promoter. They tried all that stuff. They still couldn't get it localized in high concentration. So then this kind of bypassed all of this because it produced more lycopene, which was then converted into beta carotene. So again, very smart, but someone had to, oh, let's try this, and then it worked. Okay. Yeah. No, that's fine. Very important.
Are we at all concerned about introducing bacteria and antibacterial something into the gut where we're concerned about the
Yes. Yes, we are. Short answer, yes, we are. A lot of people are very concerned. So that brings up a good point. Okay, so we're making these constructs and we're adding in an antibiotic resistance gene into all of them. And then we're consuming the product. And the companies will say there's no risk to consuming the antibiotic resistance gene. That tells us that. However, we already know that consuming chicken and other poultry and beef and pork that have antibiotic resistance genes in the bacteria in those animals translates into bringing antibiotic resistance gene into the human normal flora. So the argument from the company that says there's no risk to this is preliminary. It's not that I don't believe it, it's that I want to see the data that shows me there's no risk. I want to see X thousand number of people have eaten X thousand number of pounds of this food, and there has been no increase in antibiotic. I mean, that's the number I want, and that number is not provided. So it's either not being done or it's not being disclosed. Hopefully it's not the former. So I agree with you, because the normal flora is interrupted very easily with antibiotics, changes in diet, stress, hormone, my goodness, everything interrupts it. So if it's not replenished, then that leads us down the path of the normal flora or the microbiome. Yes. Yes, it's very big. It's a very interesting area of research. The name of a book just went through my mind and I can't remember. Maybe you've read it already. Gosh, I'll look it up afterwards. There's a book, an MD researcher just published a book last summer. I read it, and talking about that very thing, about what we're doing. We're basically eliminating our normal flora because of the way we eat and live and wash and.
It's almost the least of our worries because if you consume alcohol and sugar.
Right.
We've been doing it all your life.
We've been doing it all our life. It's the replenishing. Right. So you have to. Right. It's the taking the. We've done. Right. Right.
There's a much bigger picture.
Right, but it goes into all of that, yeah. Yeah, exactly, but it's kind of a very interesting overview. But if you're looking for technical, it's not it's not technical enough if you want real hard data. You might read that first and then read the next. You know what I mean? Yeah, I'll look it up afterwards. That's also true. We've pretty much eliminated a lot of normal flora from most humans, but not all because of the way we live. So this is a little aside. But if we compare the normal flora of, say, us, living in the Western world, no matter what we do, I take probiotics, I eat organic food, I only use vinegar, I mean, I do all of that, right? But my normal flora is still the exact same as everybody who does not do any of those things, pretty much, in Kansas, let's just say. But if we compare our normal flora and we choose a group of indigenous people who live outside Maine society that do, I mean, there's lots of groups of people that come out of the Amazon rainforest, for example. A group of an indigenous tribe came out last year that have never been contacted, we think. When we compare our normal floras, they are very different. So the normal flora of humans does change depending on our environment, which means we can alter it. We know that. There is one normal flora called nitrosomonas, which is actually normal flora of skin, and it comes from soil, of course. But we pretty much have eliminated it in the Western world because we have a shower every day. or once, hopefully, every few days. You know what I mean? And we use soap. But in other people who do not use chemical soaps, maybe they use something more natural, it does not eliminate, like a coconut-based soap or something. It does not eliminate, and they have much more decreased rates of eczema, skin allergy, skin infection. I mean, very, very radically different. It's very interesting. Of course, with the Western or pharmaceutical answer is then, oh, you can buy it. You can buy it and spray it on. It's called, it's bacterial mist, and it's expensive. This semester, one of my students upstairs, we bought it, and that was their semester project to research it. It was very interesting. It does work. It does kill it kills E. coli, and it kills MRSA. It was pretty cool. AO biome. So capital A, capital O, biome. B-I-O-M-E. You can look up their website. I mean, their website sounds amazing, but of course they all do. But so we actually tested it. I mean, it does what it's supposed to do. It was pretty cool. But anyway, that's a complete aside. But yes, so this is, I would be concerned from the normal flora point of view, and antibiotic resistance in general. Well, we started eliminating them. They're everywhere. Yes. You need to replenish your normal flora, but you could also go out and just eat a teaspoon of dirt, and you would do the exact same thing. Vegetables that you grow yourself, because there's traces of the dirt. It's bacillus species and lactobacillus. They're all in the dirt right there. Of course, you could also eat anthrax and Clostridium. I mean, so we've eliminated one problem and brought another. So, yeah. But you know what? If you're out in the gardening and you grow your own vegetables, you are already supplementing your own normal flora from the dirt. That's where it is. So if you eat that food, I mean, of course you're rinsing it, but you're not rinsing it all away. But when we eat sterile food, and our food is by no means sterile, I mean from the store, we are decreasing the amount of normal flora that we're naturally eating. Or if you make your own yogurt, if you've always eaten your own yogurt, you don't need probiotics. But if you buy Yoplait, I don't care what it says on the packet, because we've tested it, there is very few live bacteria left in Yoplait or Danone, or pick any of those ones. They're just a waste of money, and way too much sugar, actually. Yeah, way too much sugar. So you need to buy what I call a real yogurt, which is harder to find, probably more expensive, and you could make it yourself for a lot cheaper.
And we are so hard on our bodies, the things we control or can't control.
Right.
And all that it does give the organs in the body half a chance of functioning somewhat.
Right.
The way they're supposed
to.
So you do empower it by consuming.
Oh, yes. Right.
You don't want to just say, okay, well, no, don't take probiotics.
Right.
Yeah.
There's nothing wrong with taking them. Right, right. And there's nothing wrong. Oh, yeah. I'm fine with taking probiotics. I take them, but I don't take them every day. If I haven't eaten yogurt, I'll just take a probiotic. I mean, it's fine. I think it's self-therapy. I make myself feel better. Especially with 3 children in the tummy bug. I'm like, I know. I'm like, oh, my gosh. I've told their teachers, you tell me straight away if there's a tummy bug in the classroom. I don't want to find out the hard way that night. I want to be prepared. Oh, my God. There's nothing worse, as you all know. Anyway, okay. Oh. Anyway, so back to the golden rice. Okay, so there was 2 beta-carotene genes inserted, and then the lycopene gene. The beta-carotene, one came from the daffodil, Narcissus, and then one came from a soil bacteria, Erwinia. That was a carotene desaturase. The daffodil one was a phytoene synthase. So both of these are precursors, that these genes make precursors to beta-carotene. And then the tomato gene was lycopene cyclase, okay? So they initially inserted that because they thought that would also be required and the 3 genes would work together. But then after the fact, they found a different type of wild rice that already had that gene, so they didn't have to use that after the fact. So they inserted so the group of genes worked together. Now, a narcissus, a daffodil, that's just a plant gene. That's no big deal. The gene from the tomato, that's no big deal. Erwinia viridora, there are some Erwinia species that cause human disease. They're not unknown. I wouldn't call them rare, but they're not common. But this Erwinia species does really not cause human disease. It would want to be an extremely immunocompromised person exposed to a very large amount of it straight into their bloodstream. I mean, very rare circumstances. And it's using a gene, not the whole organism. So there was concern about using, because of course it was like, we're using an Erwinia gene, or Erwinia causes disease in immunocompromised people. So again, it's the balance of knowledge. Yes, we have to be careful using bacterial genes, but we're choosing one gene, and it's a precursor for beta-carotene. So the genetic design, the construct that we talked about earlier, is a pretty good construct in terms of safety, and it works. So the genes were transformed, so they were inserted into the cell, into the nucleus where the DNA is. They were put under control of a promoter. So we talked about the promoter at the beginning that controls turning on. The promoter that they inserted into the plasmid was endosperm specific. So that means the promoter only worked in the endosperm, which is the part of the rice we eat. So that's why we're only getting beta-carotene in the endosperm, not in the rest of the plant where we're not eating it. So the pathway here, so the bacterial gene that they use to complete the pathway is involved in, so the lycopene gets produced by the CTRL gene. Then we have the precursor genes for beta-carotene. So lycopene gets converted into beta-carotene, and then that builds up and is expressed in the endosperm. Okay. So the plant enzymes process the lycopene to the beta-carotene in the endosperm only, and that's what gives it the color, because beta-carotene gives yellow-orange color. So that's why it was called golden rice. They improved the version in 2005. It was released called Golden Rice 2. They added in another gene from maize, which is just basically a variety of corn. So they added in another phytoene synthase. This produces more beta-carotene. So they basically increased the amount of carotene by adding in another gene. The recommended daily allowance for beta-carotene in terms of how much we would need to eat, we would have to be 144 grams of rice. The highest yielding rice would have to be consumed to actually give enough final product vitamin A. Okay? So the availability of carotene from golden rice has been confirmed, and it's more important, it has to be bioavailable. So bioavailable means that when we eat it, we can actually use it. Okay? So it has to be in a form that humans can digest and that can use the beta-carotene to make their vitamin A. Okay? Some of the early studies used precursors to beta-carotene that humans were not able to use, so that the early strains were not of any use because it could not be used to make vitamin A. That was one of the early problems with it. And that early issue is, anytime you hear discussion in the media about golden rice, that is always brought up every time. Someone always says it, but we can't even use the beta-carotene that's in golden rice. Well, we can. It was fixed. They chose the wrong one. Not very smart. But now it is bioavailable. So we know that a person can eat the golden rice. The beta-carotene in the rice can be used to produce vitamin A in the human using their own conversion enzymes. Okay. So the next, following on from this, the researchers that, the last round, the researchers produced the Golden Rice 2. They received further funding. The recent funding came from the Bill and Melinda Gates Foundation. And this foundation funds a lot of this type of activity, humanitarian, global activity. They fund a lot of public health projects, vaccination projects, supplementation in terms of nutrition, education for girls. They really do a lot of public health projects globally. So currently what they're working on is they want to increase bioavailability, so further increase the beta-carotene to give the vitamin A, vitamin E, iron, and zinc, and also to improve protein quality through genetic modification. This is a tall order to get all of this into rice. I have no doubt they can figure it out, but it will probably take another few years. Some small issues. Vitamin E is fat soluble, so you would have to be very careful that there wasn't too much vitamin E, because you store vitamin E, and that can lead to vitamin E toxicity. So they're going to have to be careful about that. Zinc is fine. I mean, zinc is a trace element, so they probably won't add too much zinc in there. Iron, you need more iron, but I don't think that'll be a problem. Protein, I'm not really sure how they're going to improve protein modification. If they start adding in soy protein, that might cause an issue. But there's not a lot of information, there's not a lot of information yet what they're doing here. So it's obviously still in trial, and they're still trying to do it. But if it could work, it would be nutritionally very, very beneficial. Okay. Now, the vitamin A deficiency, so that was the original goal, to produce rice to supplement for vitamin A deficiency. So I talked about the deaths earlier, but in terms of blindness, it's responsible for overall has been attributed, so in that we have collected data for 1 to 2 million deaths and 500 cases of irreversible blindness. Children and pregnant women are at highest risk. And right now, the World Health Organization has 122 countries on their list of countries that are affected by vitamin A deficiency in terms of how many people. At 2005, we don't have 2020 numbers yet. You know, they do it kind of every 10 years. But the last big group of number that they reported was 2005, and that was just one time point, so that was a prevalence, not an incidence, it was just a prevalence. So at that time, that's what they estimated, 190 million children and 19 million pregnant women in 122 countries were affected at that one point in time. So the numbers are staggering. Now, the benefit, the countries where there is the highest amount of vitamin A deficiencies, a lot of these countries would be countries where we could grow rice anyway, and rice is a staple food in a lot of those countries. So it is helpful to be able to grow the food in the locality where children are suffering from vitamin A deficiencies. So that would make sense. So putting in the precursor, the beta-carotene precursor, has been really a less expensive alternative just to vitamin supplementation. So there was some discussion of, you know, all this work to produce vitamin A, can't we just supplement? Can't we just give them supplements? But that was seen as more costly and also just not very process possible. I mean, we can't even vaccinate, not to mind get enough vitamin A supplements out to half of the world. So the idea was if we could produce a food product that, yes, there may be the cost up front, but that in the long term it would be more beneficial. And also just that families could grow their own food, which is socioeconomically just better for families to be able to do that. Okay. So one of the concerns was, is there enough? So is there enough vitamin A? So once they got through all the safety and we were considered that it was safe, are we providing enough? Initial analysis of the nutritional benefits of rice suggested that the consumption, so the round one, version 1, would not eliminate the problems of vitamin A deficiency, but should be seen as a supplementation. So there was concerns with the round 1 that it was just still not enough. So that's why they worked on it to produce golden rice 2. Okay? So since then, the improved strains of golden rice have been developed, providing more beta-carotene. So they added that one more gene. And this would provide the entire recommended allowance of the nutrient who ate about 75 grams of golden rice per day. So they would be able to consume enough rice in order to provide the right amount of vitamin A. Okay. Now, beta-carotene is hydrophobic. This is another kind of part of this puzzle. So it needs a significant amount of fat in the diet to be bioavailable. So it's not. So if it's hydrophobic, it's not going to bind well and interact with water. So it binds better and interacts with a source of fat. So this assumes that there is enough fat present in the diet to be able to consume and absorb the vitamin A. So it's not a one-size-fits-all here. So we just can't say eat some rice and all the problems will go away. There does have to be some low concentration of oil, either animal or plant oil, available. So when we look at vitamin A deficiency in the developed world, you know, if someone has a vitamin A deficiency, usually it's going to be coupled with some other nutritional factor. Because that means they're just not lacking vitamin A, there's some other things lacking in their diet too. In the developing world, where they really have multiple nutritional concerns, then we're talking about a different situation. So we really can't compare them in terms of developed versus developing when we talk about vitamin A deficiency. So it's still an excellent food in terms of a nutritional supplement, and then hopefully with the added benefit of what they're going to work on, or hopefully what they're working on, it will become a better supplement in terms of nutrition. But it still requires a somewhat okay diet for a child to be able to eat, to be able to get that small portion of fat, oil in the diet. Okay. So so that's the golden rice with beta-carotene genes, pretty much. Okay. The next group of crops all have this guy in common. This is a bacteria, Bacillus thuringiensis, and it's abbreviated to BT. And you've probably heard of BT corn and BT cotton. This is where BT comes from, the name of the bacteria. So we're going to talk about BT here for a little bit. So this bacteria is another soil organism, so naturally occurring in the soil. It's gram-positive, so that would put it in with the same class as Staph aureus and other bacillus species, Bacillus cereus, and so forth. And when they sporulate, so bacillus bacteria produce spores, and that's what they use to survive harsh conditions until nutrients are available again. Okay? It's also why they're very difficult to eradicate, because spores are very difficult to clean. They're very difficult, almost impossible to disinfect against, because they're so hardy and resistant to chlorine and ammonia and all sorts of things. So when these bacteria produce spores, they produce crystal proteins, and they're proteinaceous inclusions, so they're proteins, and they're gathered together in little vesicles, so that's why they're called inclusions or inclusion bodies. And they have insecticidal action. Now, in the natural world, you know, they're living in the soil, they're in contact with insects. When they sporulate, they produce these proteins that kill insects. Okay, so over time, they have evolved to live in this environment. Okay, a lot of bacillus species of bacteria associate with the roots of plants. So you find a lot of them on roots of plants. So over time, this has developed as a symbiotic relationship. Bacteria grows on the plants. It uses the nutrients from the roots in terms of nitrogen would be the big one. And then it produces a small amount of a protein that kills insects. So that would have been beneficial to plants and to the bacteria. Okay, so in the natural world, that's how this would have happened. Okay, we, however, have come along and figured out, whoa, this bacteria makes a chemical that kills insects. Let's see what we can do with it. Okay, so that's how it started. It originated as an insecticide, this protein. So the proteins are called cry proteins after crystal. Okay, they're usually pronounced cry, not cree. The cry proteins are encoded by the CRY genes. So these are the genes that produce the proteins. In most strains of these bacillus bacteria, these genes are located on a plasmid. So we're back to another plasmid construct, which means then that we can take the plasmid and move it and bring along the CRY genes with it. Okay? So they're already on a plasmid, very handy for us. All right? So they're not in the chromosome of bacteria. They're on a plasmid. Now, originally, these proteins have been used to produce sprays, insecticide sprays, and they were since the 1920s. So this is not a new development. It was recognized that these bacteria produced the insecticide protein, and they grew the bacteria, isolated the protein, and used that to spray as an insecticide, and it worked. Okay, the insecticides are still used today. The two big ones are sold as Dipel and Thuricide. So they are still used as just straight spray-on pesticides today. The manuals for these products, you do contain warnings in terms of environmental and human health warnings, and they contain very specific labeling about how much concentration should be used over what area, and to use personal protective equipment, much like all pesticides and insecticides. So, but we still use them. The most recent review of this chemical was in 2012 by the European Union in terms of a safety review. And this review panel reported that there was data to support some low toxicity to humans and the environment, but there was not enough data to justify taking it off the market. So it still is legally sold insecticide. Okay, so some of the media discussion has been that it was claimed not safe or it was taken off the market when they talk about BT corn. The review panel did not say it should be taken off the market, but they did say that there was low toxicity issues, but not enough to remove it. Okay, so the first one they produced was BT corn. So this has those genes, those crystal genes, inserted into the actual corn genome. Okay, so the corn then produces the protein, that CRY, that crystal protein. They've called it delta endotoxin, and it is lethal to European corn borers, which is a common pest on corn plants. So the idea was that the corn would produce its own pesticide, and then when the insects came and tried to eat the corn, they would consume the toxin and would die. And that's what it does. So the corn produces its built-in own pesticide against the corn borer, which is the most common pest for corn. So farmers who plant BT crops then do not have to apply an extra pesticide to kill these insects because the corn itself is killing the insects. The first set of experiments using this in a plant against an insect was on a silk farm, and it killed silkworms. Okay, so they kind of tested it on silkworms, and it did kill them. And they're in the same genus as European corn borers. So someone thought, well, let's see if it kills corn borers, and kind of went from there. So the company that originally designed it or did this study was Plant Genetic Systems. They're from Belgium. Now they're part of Bayer Crop Science, which owns half the world, it looks like. And that was the first company to develop genetically modified crops in 1985. They did their first research on tobacco. So they took the genes, they put them into tobacco, and they tested it in tobacco. This BT tobacco was never commercialized. They used it as a test crop. So they tested out the process in tobacco to see would it work, and then they went ahead with the corn. They use tobacco plants because they're very easy to grow, manipulate in the lab, and they're not part of the human food supply. So there was no risk if something went wrong that it would end up in the human food chain, because they are in no way, in any way, part of the human food supply for any reason. Okay. So they started with that initial BT, and we have seen insects develop resistance over time. Okay, so what they have done, just like we do with antibiotics, every time we see significant resistance to the BT toxin in the corn, they have developed new strains of the BT to produce a slightly different toxin, and then they use that. Each new strain is given a unique number, an identifier, and is registered with the EPA. So the EPA does list all the ones that are approved, and the formulations of ones that are approved for farming in the United States alone, for farming for human food production, as opposed to animal feed. They do list those. It's the OMRI, the Organic Materials Review Institute. So you can go on there and look at the formulations of BT that are provided for human use, and the EPA registers all of them, ones that could be used for human and animal. Okay, so you can kind of find them in two ways. So corn, the BT corn that they have used produces that protein, okay, and the protein is poisonous to the insects. The European corn borer, prior to using the BT corn, caused about $1 billion in damage to corn crops every year. So it was a significant source of lack of revenue. In recent years, there have been traits added to the BT corn to do also corn earworms and root worms, because once the European corn borer was killed off, then of course new insects came in that kind of took over their spot. The root worm also causes about $1 billion in damages, so they added traits into that one to kill those ones. The BT protein is expressed right throughout the plant, from the leaves into the root, okay? And when the insect comes along and eats part of the plant, the protein is consumed from the plant and then goes into its intestine and is activated in the intestine. In the gut, the intestine of an insect is alkaline, so it's basic, high pH, okay? So like it's probably 9 to 11, and that activates the protein. The human intestines are acidic. We're like 3 to 5, maybe a little lower. So our intestines are acidic. So our stomach acid will not activate this protein. So that's why it's not directly toxic to us, because our acidic stomach will not activate it. It requires a basic pH to activate, right? So in the alkaline environment in the insect, the protein unfolds in the alkaline pH. It's cut up, it's cleaved by other proteins. It forms a toxin, so then it becomes toxic. It paralyzes the digestive system and pokes hole in the gut wall, and then, of course, the peritoneal cavity, much like what would happen in a human, breaks open, and then the insect stops eating and then dies because they can't eat. So it kills the insect within a few hours. Very efficient. Okay. The EPA requires farmers who plant BT corn to plant what they call a refuge. This is a non-BT corn field nearby to provide a location to harbor vulnerable pests. Because, we'll talk a few specific examples of insects, because of course this toxin can also target other insects who are not a threat to the crop. Okay, and all insects have an important part in our ecosystem, no matter how much we don't like them. Right? So if we just eliminate insects out of hand, then we have problems with pollination and decay, like all the flies that are involved in the decay system. So the EPA says that farmers have to plant a refuge. The theory behind this is that we're providing an area of non-BT crop where these insects can also live to try to slow down the development of resistance in other insects that we're not concerned about eliminating. Now, the EPA also requires companies to train farmers how to maintain refuge, how to collect data on the existence of refuge, and to report that data to the EPA. So the companies that provide the seeds are supposed to do this. Okay? You can imagine that it probably may or may not happen. At the beginning, the first two reports were very good. Between 2003 and 2005, compliance with keeping refuge was above 90%. So farmers really did plant some non-BT species to allow for this refuge. But by 2008, only about 25% of corn farmers did not keep refuges properly. And since then, it has decreased even further. There's also other problems in terms of nothing really got to do with the EPA, but how the seed companies are doing business, that they do place a lot of pressure on farmers who do not plant BT corn or the other ones that we're going to talk about, so that gradually over time the number of farmers who do not grow these seeds are very, very minimal. And then because if they want to produce their food to be non-GMO, they can't use the same hauling trucks, They can't use the same grain elevators. They can't use the same processing plants. So it makes it harder and harder for them to be able to get their product to market because they have to ship it longer and longer. And then a small farmer just cannot afford that. So some of the companies who do produce non-GMO crops have started like either assisting small farmers or just buying the small, just helping them with the process to try and get enough grain that's GMO negative to then produce GMO crops. But for a few years, there were a lot of small farmers just going out of business because they had no way to actually produce their corn once they grew it, because all the big companies didn't help or didn't assist really. Yes. Yes, it has to be, there's a ratio compared to how much of the BT they're growing. It is a ratio that you have to have so much per so much. They don't have to use it, or they could, but if it's grown on a BT farm, it won't go, you know what I mean? They don't use it because a lot of it would be eaten up by the pests, or they would just mow it down and then till it under for next year. So in terms of benefit, between 1996 and 2007, there really was a good control of the pest populations once they initially introduced the BT corn, because it really did eliminate a lot of the pests. So initially, it was seen as a really positive stride that a lot of pests were eliminated, and that also was a benefit for other crops, whether they were GMO-free or not, because it eliminated crops from the area. But after that, then resistance started to trickle in, and we had problems. So we'll look at some of that. So resistance started, and it developed in areas with either defective or absent refuge management. And that makes perfect sense, because if the whole area is covered in the crop that produces the toxin, eventually the insect is going to develop resistance to it, just like bacteria do, just like viruses do. It just happens. You have millions of pests out there. Some of them are going to genetically modify themselves to be able to tolerate the toxin. That's how it happens. In 2009, Monsanto, which is one of the big companies that grows these plants, found a pink bollworm in India. And it had become resistant to a BT cotton. We'll talk about that in a little while, but it is a CRY gene, similar gene. This was the first incidence confirmed by Monsanto globally in 2009, even though prior to that there had been a lot of discussion about it, that they were seeing it in the fields, but that was the first real announcement by Monsanto. This resistance to cotton has been since seen in Australia, China, Spain, and the United States. In 2012, in Florida, another group of insects, armyworms, were found resistant to pesticide-containing corn, GM, genetically modified corn. This particular species of worm had been seen in Puerto Rico in 2006. So we do have resistance issues with this scenario, okay? So a lot of concern was on these non-target insects. So we've got all these other insects that are very important for lots of other things. And are we killing them off because we want to kill this one pest? And what's that going to do? So there have been claims, and important ones, that these proteins could target other insects as well as the ones that they wanted to. Now, there are both sides to this. These proteins, in terms of using them as a spray and not in the corn, have been used in France since 1938 and in the United States since 1958. There has really been no major reporting, no big reporting in terms of how much damage they are causing to all pests when they are used correctly, okay? But that is not the same scenario as putting them in the plant itself, because if you spray the pesticide, you spray it on the leaf of the plant. Some, of course, might get into the soil, but you're not putting it into all the plants in the whole area. It's going directly on the plant. It lasts for so many, it has a finite half-life, and then it's gone. It breaks down in the sunlight. So it's not, we can use that data, but it's not a fair comparison, okay? Some of the CYT toxins are toxic to other insect groups, the beetles, some flies, and some of them are, and this was a big issue, some of them do target some moths and butterflies, which of course is concerning to a lot of people. Beetles and flies, even though we mightn't like them, they're very important in the breakdown process to break down the detritus, to break down leaves and the humus and all that organic matter. So we really can't be targeting those guys because we need them to break down the soil, okay? And of course, the moths and butterflies we also need for various things.